All right, so these are your section 5 notes for chapter 2. This section is all about enzymes. So your objectives for this section are to explain the effect of a catalyst on activation energy, but also to describe how enzymes regulate chemical reactions. So we're going to talk about enzymes and catalysts. Ultimately, we need to understand that enzymes are catalysts, right? So we could interchange those words, right? But enzymes specifically are proteins that act on catalysts inside of your cells. So it's all about catalysts, all about enzymes, and how do those help your cells function. So first off, activation energy. This is actually an old definition. It was from last section. We just need to touch on that again so that we can further explain how catalysts affect these reactions. So I need to draw an energy curve again, just like last section. It wouldn't matter if it was exothermic or endothermic, so I'm just going to draw an exothermic reaction here. Right? And you have to remember, you always have to go up before you can come down. So this is just like last section, shouldn't be an issue. I have this activation energy up here that I have to reach before that chemical reaction is going to start. And the reason why I needed to add that energy to my system was so that I could break the bonds in my reactants, because then once that activation energy is reached and all those bonds are broken, I can form new bonds in my products to finish things off, right, to complete that reaction. So again, I talked about before how you're riding a bike up that hill. It takes a lot of energy to get started, but once you get to that activation energy, once you get to the peak, the apex of the hill, you can coast just the rest of the way through. Right, so we needed to add that energy to get started, but that's it. Right? It goes through to completion after that. Now, specifically, because this was exothermic, I'm releasing more energy than I'm absorbing, but we're focusing on that absorbing part right now. So that red arrow, how much energy did I need to start? So then a catalyst, right? A catalyst is something that helps lower activation energy for a reaction. So if I draw that same picture again, you're going to need this drawing in your notes, so make sure you draw it again. Again, it's exothermic. I could just as easily do this with an endothermic reaction, but a catalyst is going to lower my activation energy. Right now, my activation energy is up here. That's the amount of energy I need to add to my system so I can break the bonds of my reactants to form my products. What a catalyst is going to do for you is it's going to lower that activation energy. So instead of needing to get all the way up to this current line, I'm going to lower that activation energy, so I only really need to get to here. Maybe. I just randomly chose a new energy level, but it's lower than that original activation energy. So now I'm going to have a different curve that goes with that. So I'm still going to start my reactants at the same level, and my products are going to end in the same place. But because there's a catalyst present, I'm not going to need to add as much energy to start this reaction before it will go through to completion. All right, so you need a picture that looks a little bit like this. You need to probably use a dotted line since you might not have a blue pen available for you. But either way, that new line you need to differentiate somehow, maybe highlight it, whatever you need to do. But you need to have an appropriate label. You need to understand that that's my energy curve with the catalyst versus my original line, just that black line, that was without a catalyst. So without a catalyst, I had to get all the way up to here to start my reaction. But with the catalyst, I lowered that activation energy and I didn't need to add as much energy to my system to get things started. So it's really, really useful for your cells. Right? It's really useful in general because sometimes you don't have that much energy available to start your reaction. All right, and there is that whiteboard question down at the bottom there. Ask what effect does a catalyst have on activation energy? That is not supposed to be difficult at all. The answer is that it lowers activation energy. It makes it easier for that reaction to start. So just make sure we drew that picture here to try to understand that, right? Put that also in words. That catalyst is going to lower the activation energy so it's easier for you to start the reaction. Right? It doesn't take as much energy to get that reaction going. So then we put that in context with your cells, right? In your cells, you have enzymes. So this word right here, we're defining that here in a second. But an enzyme is a catalyst. It's just specifically a protein that acts as a catalyst inside of a living thing. So why are those enzymes, why are those catalysts important to living things? Now this is also a whiteboard type question, but because it's very difficult to put it into words, I'm just going to give you guys a good answer for this. All right, so the whole idea here, glucose burns at 300 degrees Celsius or 572 degrees Fahrenheit. That's really, really hot. So if I want to burn sugar in the lab, I need to heat it up to that temperature before it'll ignite. That's my activation energy. I have to get it to that high temperature and then it'll burn in the presence of the oxygen in the room. But since you need to burn glucose in your cells at a much lower temperature, only 37 degrees Celsius or 98.6 degrees Fahrenheit, you're going to need an enzyme to accomplish this. Since you're not the Human Torch, you can't get all the way up to 572 degrees Fahrenheit and survive that. So you're going to need enzymes because your cells have controlled conditions. Right? So we're going to put this into words. You need enzymes and or catalysts to allow reactions to occur in the controlled conditions of the cell. Right, so since you have to maintain homeostasis, since those cells have specific, very specific conditions, you're always going to need an enzyme for reactions because you can't increase the energy significantly. You can't be the Human Torch to burn that glucose. So you're going to always need those enzymes because they allow all those different reactions that are supposed to occur in those controlled conditions. You don't always have the energy necessary to get things started just regularly, but with an enzyme, with that catalyst present, you can lower that activation energy enough that those reactions can happen anyway, even without that extra energy. So this is just a stock answer, right? You can put it in your own words, but just make sure that you understand that enzymes are really, really important inside your cells. Okay, so this next slide, I have these next two definitions already uncovered, but enzymes are proteins that act as catalysts for reactions in living things. I just told you that an enzyme is a catalyst. It's just specifically a protein that acts as a catalyst in your cells. And then we're also going to have this term substrate. Substrate is specific to when you're using that word enzyme, right? But your substrate is actually another word for reactant. But it's a reactant that fits into your enzyme and that enzyme is going to help that reactant turn into some product. And it fits into that enzyme just like a key in a lock. So that's where we come to this first model, the lock and key model. So I need to draw something here, and you need a picture just like this in your notes. So this is going to represent my catalyst, right? My enzyme. And notice that enzyme has a little notch in it. And that notch is supposed to perfectly fit the substrate, the reactant that goes with that enzyme. So this enzyme is supposed to act on this substrate, so that substrate fits into that enzyme just like a key in a lock. Excuse me. Because I need to turn this substrate into a product, but I can't do it without this enzyme. So I want this substrate to turn into some product over here. I'm going to draw my product as a box, just random arbitrary decision there. But it started out as a triangle. It's going to change into that square shape over there. But I can't do that directly. I'm going to need an enzyme to help me change that reactant to a product. So that's what this model's for. So I'm going to make this enzyme substrate complex first. Right, so that substrate fits into that notch. And with the help of the enzyme, I can make my product. But without the enzyme, it would not have been possible. Right, so again, enzymes are important in your cells. You want to change that reactant to a product, that substrate to a product, but without the enzyme, that's not possible, which is why this model is really important for your understanding. I'm going to need this enzyme substrate complex in the middle in order to make my product. And this enzyme is specific to this substrate, so only that specific reactant fits into this enzyme so I can make a product. When it's all said and done, and you do form your product over here, that enzyme is actually going to be conserved, right? It's going to release that product once it's formed, and it's going to go back to the beginning. I'm going to find another substrate, another reactant, and try to turn that into a new product. So it just loops back to the beginning. But you're going to need that enzyme substrate complex in the middle here in order to make this all work. Right? That reactant can't turn into the product directly. You need the help of the enzyme. So this model's good, but it turns out there's a better way of explaining how those enzymes affect your substrates, how they affect your reactants, and that's why we have this next picture, the induced fit model. So some of the parts are the same. I'm still going to need an enzyme, and it's still going to have that notch that perfectly fits my substrate. The difference between this model and the last one is this one's trying to explain exactly how that enzyme affects that substrate, how it helps it become your product. So when I draw this enzyme substrate complex, I need to see a change. I need to see a difference in that substrate. So from that triangle shape, once that fits into the enzyme, what's going to happen here is I'm going to have a change in that shape. Right? It's called induced fit because once that enzyme and substrate come together, that enzyme's going to change the shape of that substrate so that it has a higher energy and it can more easily become the product that you want to form. So this is a basic chemistry concept that I know is a little higher than what you really need to know at this point, but all molecules, no matter what they are, always have a specific shape, and that specific shape is related to its energy. They take on whatever shape has the lowest energy possible. So if that substrate is a triangle, it wants to be a triangle. That's a low energy state for that substrate. But when the enzyme forces that triangle to look more like the circle, when it changes the shape of that substrate, it actually increases the energy of that substrate. So you need to understand that with this induced fit model. In this enzyme substrate complex, I'm going to change the shape of that substrate because changing the shape is going to increase the energy of that substrate. It doesn't want to be that circle shape, it wants to be the triangle. So when you force it to be that circular shape, that changes its energy, it increases the energy. And now that it's in that higher energy state, you can more easily make your product. Again, we just arbitrarily picked that square as my product shape, but it really wouldn't matter what it was. But now it's a different thing. I use a different color because now I have my product as opposed to my reactant or my substrate. I've changed that into a product. And again, that enzyme's conserved. It's going to go back to that original state where it's got the notch that fits the substrate. It can again go back and find a new substrate to change into a product. But this one is better able to explain how those enzymes really work because you need to change the energy of that substrate somehow. And you're going to do that by changing the shape. So with those two models, we can explain how enzymes work. All right, so here's another whiteboard question, and this is a tough one, right? We just talked about how that enzyme doesn't just meet up with the substrate, it has to change that substrate somehow. All right, so that induced fit model better explains how enzymes function because it helps you understand how the energy changes. All right, so you could put this answer into words, but I'd really actually prefer to just draw it and help you and see it that way. So I have a couple different things that I want to look at here. This is an energy curve again. You can make it exothermic. It could have just as easily been endothermic. But I have a certain activation energy. And I'm trying to explain how those enzymes function. I'm going to change that activation energy in a different way. So we started out with a substrate over here. I had a reactant, that triangle, that I want to turn into a product, the square. And in order to do that, I use an enzyme. And that enzyme changes the shape of my substrate to increase the energy of that substrate to help me turn it into a product. So I'm going to change my activation energy, but I'm not going to do it the way that we did it before in that first picture. A more accurate representation of this, if I'm going to change that the substrate, what I'm going to do is I'm going to increase the energy of my reactant so that I'm up here instead of where it was before. And then that new shape, we used a circle for that. Since that new shape has a higher energy, the result is that I got a boost, right? I'm now closer to that energy that I need to start the reaction. So instead of lowering the activation energy, we're thinking about this as changing the energy of the reactants to effectively lower that activation energy. So I need a second curve. Again, if you need to use a dotted line, you should use a dotted line. But the change in shape caused this increase in energy for my reactants. So when I change the shape from this triangle to the circle, I had an increase of energy I went from down here to up here, so I got that boost. And because of that, I needed less energy to start my reaction. Now I only have to add this much energy to break my bonds as opposed to this much energy to break my bonds. So I effectively lowered my activation energy by changing the energy of my reactants by giving them a boost.